Hello, my name is Briana Barnum, and today I will be presenting a paper called Incorporation of Pseudouridine into mRNA Enhances Translation by Diminishing PKR Activation. And this paper is by Kariko and Drew Weissman. RNA modifications are widespread in eukaryotes and influence RNA stability, structure, and function. Pseudouridine was the first discovered RNA modification and is found in mRNA, tRNA, ribosomal RNA, and long non-coding RNA as well. Pseudouridine synthetase makes pseudouridine from uridine by shifting the ribose sugar attachment point from the nitrogen in uridine to the carbon 5 in pseudouridine. Unmodified in vitro transcribed mRNAs activate immune sensors such as toll-like receptors and protein kinase R, or PKR, leading to reduced total translation and cellular apoptosis. PKR is activated by binding double-stranded RNA and viral RNA. PKR then phosphorylates itself and IF2 alpha, making IF2 alpha associate more closely to IF2 beta, thus reducing ternary complex recycling and translation efficiency. Pseudouridine mRNAs stabilize RNA and reduce immune activation. In a previous study, Kariko et al. Observed that the transfection of pseudouridine-modified mRNAs enhanced translation efficiency, but they did not know how pseudouridine-containing transcripts interacted with PKR. The aims of this paper was to assess the translation efficiency of pseudouridine-modified and unmodified transcripts, investigate how pseudouridine-modified mRNA interacts with PKR, and to determine if pseudouridine-modified mRNAs reduce PKR activation and IF2 alpha phosphorylation. Translational repression is induced by in vitro transcribed mRNAs. In image A, human embryonic liver cells were co-transfected with two luciferase mRNAs that were unmodified or pseudouridine modified. When both luciferases contained pseudouridine, translation was at a maximum. However, when only one contained pseudouridine, translation was decreased by half. This is because in vitro transcribed unmodified mRNAs elicit global translational repression. And finally, when both contained unmodified mRNA, translation of the transcripts was lowest. In image B, we can see that cells were transfected with either unmodified or pseudouridine modified RNA. Cells were incubated with 35S methionine and the proteins were precipitated. The graph basically shows that translational efficiency is increased when the transfected mRNAs contain pseudouridine modifications. In vitro transcribed mRNA stimulates PKR activation. PKR was incubated with a labeled phosphate ATP and with non-modified or pseudouridine-modified luciferase mRNAs with either a 5' triphosphate or a cap mimicking bacterial and eukaryotic mRNA conditions. The reaction products were separated by STS page and visualized using phosphoradiography. Results show that the 5' triphosphate did not have a great impact on phosphorylation of PKR, but the modification of mRNA did. PKR was activated to a lesser extent when the transfected mRNAs contained pseudouridine. PKR is not activated by pseudouridine-containing mRNA in cells. In Part A, mRNAs with or without pseudouridine were placed into cells. Scientists then tested the activity of intracellular PKR by doing a Western blot for PKR phosphorylation. Here we can see that PKR phosphorylation was greater when the cells were treated with non-modified mRNAs. In Part B, a Western blot was used to visualize the phosphorylation of IF2 alpha in cells transfected with either unmodified or pseudouridine modified mRNAs. The results show that IF2 alpha is phosphorylated less when transfected mRNAs contain pseudouridine modifications. Suppression or loss of PKR enhances unmodified mRNA translation. In Part A, plasmids of CAL, K3L, and 2K3L mutants were transfected into human embryonic liver cells. CAL and K3L are viral proteins that inhibits PKR. Cells were then transfected with unmodified or pseudouridine-modified firefly luciferase mRNAs, and luciferase activity was measured to represent the fold increase in translation. Here we can see a greater increase in the translation of pseudouridine-containing transcripts when PKR was inhibited. Graph B shows the fold increase in translation in wild-type and PKR knockout mouse embryonic fibroblasts transfected with non-modified or pseudouridine-modified mRNAs. There was a greater fold increase in the translation when cells were transfected with pseudouridine. However, in PKR knockout fibroblasts, there was no difference in the translation between modified and pseudouridine-modified mRNAs, confirming that translation differences between the mRNAs is due to PKR. Image C shows IF2 alpha phosphorylation in wild-type and PKR knockout cells. Pseudouridine transcripts cause less IF2 alpha phosphorylation than unmodified transcripts in wild-type cells. However, in the absence of PKR, IF2 alpha was not phosphorylated by either mRNA, showing that PKR is the link between in vitro transcribed mRNAs and decreased translation efficiency. PKR doesn't engage pseudouridine-containing mRNA. In Part A, scientists wanted to determine if pseudouridine-modified mRNA was a competitive inhibitor of PKR, so they mixed 200 base pairs of double-stranded RNA known to activate PKR with varying excesses of pseudouridine-modified firefly luciferase mRNAs and incubated them with PKR. The products were visualized on an SDS page. We can see that pseudouridine-modified mRNA failed to inhibit PKR activation by double-stranded RNA. In Part B, modified or unmodified biotinylated mRNAs were incubated in cells. RNA was pulled down using streptavidin beads. SDS page was used to separate the proteins, and PKR and poly-A binding protein were detected using Western blotting. Results showed that poly-A binding protein bound equally to pseudouridine-containing and unmodified mRNAs. However, PKR did not bind to pseudouridine-containing mRNAs as well as it did to unmodified mRNAs. Therefore, the reason why translation of pseudouridine-containing mRNAs is greater than that of unmodified mRNAs is because pseudouridine-containing mRNAs do not bind strongly to PKR, thus leading to PKR's weaker activation of IF2 alpha. In conclusion, pseudouridine-modified mRNAs bind PKR weakly, reducing PKR activation and IF2 alpha phosphorylation. This results in higher translation efficiency and reduced immune activation. Modified mRNAs are promising vectors for transferring genetic material. Since this paper has been published, other RNA modifications have been explored for enhanced translation and reduced immunogenicity. Some of these are N1-methylpseudouridine, which is used in an mRNA COVID vaccine, which encodes the entire COVID spike protein. High immunity efficacy has been reported for those who have taken the modified mRNA vaccine rather than the unmodified. Another modification is 1-methylpseudouridine mRNA transcripts that are used in cancer immunotherapies. These modified mRNAs encode T-cell interacting antibodies, helping to recruit T-cells for tumor destruction. Finally, the long-term stability and safety of modified RNAs will continue to be investigated. These are all of the papers that help make this presentation possible. Thank you.